Hey guys, so I'm going to record chapter 14 lecture for you. This is all about RNA and protein synthesis. Alright, so in lesson 1 we're going to talk about the types of RNA. So first of all, let's just get a quick refresher about DNA. We know that DNA is double-stranded and it has that sugar phosphate backbone that contains the sugar called deoxyribose. And it also has thymine, and it pairs with adenine, right? Okay, so those are the nitrogenous bases that they have in common, A, C, G, all right? But in RNA, RNA is not going to have T, it's going to have U, uracil. So that is one main difference. The other difference is that it is single-stranded, and its sugar is going to be ribose. Ribose is the sugar instead of deoxyribose for DNA. So U replaces the T. So when you're doing pairing, or when you're, later on we'll talk about messenger RNA, it will not be A and T, it'll be A and U. Okay? So if you see a U, you'll put A. All right. Okay, so again, the sugar in RNA is ribose. RNA is single-stranded, not double. And it contains uracil in place of thymine. So those are the three main differences. All right, what are the three types of RNA? We have messenger or mRNA, we have ribosomal RNA, and we have transfer RNA. All three of those have a very important role, and they work together in order to have transcription and translation complete and to create proteins. So here is messenger RNA. An mRNA molecule is a copy of the portion of DNA that will be used to make a protein. So here's our trusty old nucleus. And after being made in the nucleus, the mRNA will travel into the cytoplasm where protein synthesis happens. So those ribosomes that you saw, those free ribosomes that are in the cytoplasm, those are the ones that we saw when we were doing the cell project. Those are technically the mRNA or the ribosomal RNA. Okay, so this mRNA goes out into the cytoplasm to find those ribosomal RNAs. Let's get back and go to the next slide. Okay, so ribosomal is where protein synthesis occurs on the ribosomes, which are made up of two subunits. These subunits consist of several molecules of ribosomal RNA and as many as 80 different proteins. And then the third one is tRNA, transfer RNA, and it usually comes into play last. Well, T and R kind of work together. During protein synthesis, the transfer RNA molecules carry the amino acids from the cytoplasm and help and bring it to the mRNA. Okay, so RNA synthesis is also called transcription. So we start with transcription, then move to translation. So transcription is when segments of DNA serve as templates to produce complementary mRNA molecules. So we use RNA polymerase, here's RNA polymerase, in order to make that RNA strand. There's our DNA, and we use RNA polymerase to make that RNA strand that we need the mRNA to leave the nucleus then, right? Okay, so there's RNA polymerase, and that is the replication fork. So RNA polymerase unzips it and allows the mRNA to be made right there. Okay, so RNA synthesis has promoters. So RNA polymerase binds only to the regions of DNA that have specific base sequences. These regions are called promoters. So a promoter is where it binds and starts the process of replication. All right, so RNA editing. New RNA molecules sometimes require a bit of editing before they are ready to be read. So what we see here, okay, these are the introns that are being removed out in order, they're not needed, they're parts that aren't needed, so they will be removed, and then you've got the exons that will then splice together. Oops, you can't see that, can you? Okay, those exons will splice together. All right, so on to lesson 2. Let me see if I can just make this window bigger. Why won't it? Okay, I'll just move it up. All right, so the genetic code, lesson 2. The genetic code is read in 3-letter groupings called codons. Codon, codon, codon. See them? A codon is a group of 3 nucleotide bases in messenger RNA that specifies a particular amino acid. So this grouping of 3, these codons, make up one amino acid. AUG, AAC, and UCU. All right, so there are 64 possible 3-base codons in the genetic code. Some of the worksheets that I will give you will not have this pie-shaped one. They will look a little different for you, but ultimately they all do the same thing. All right, so here is our genetic code table. This chart shows us our genetic code, but it shows them to us in codon formation. All right, so if we look at UCU, that would be serine. So you start in the center, and then you work your way out. CUC, that would be leucine. CAC, that is histidine. So you start in the larger center, and then you move to the next row out, and then further out. And as you can see, some of these codons will have a different base at the end, but will still code. So the proline, for example, has 4 different combinations of codons that it can have. Okay, so again, start at the middle of the circle with the first letter of the codon, and you move outward. Boop, boop, boop. CAC, histidine. Okay. The methionine codon, AUG, serves at the start codon for protein synthesis. There are 3 stop codons. Okay, AUG is methionine, and that is a start codon. And then we've got stop here. There's 2 combinations for stop. And there's the other combination, UGA, for stop. So that's when the protein will be starting and stopping. Okay, so now we are talking about something fancy. All right, so we already did transcription is when the mRNA is being made. So transcription has already happened at this point, and now we are looking at translation. So transcribed mRNA, there it is, directs the translation process. Translation is the process that produces the proteins by decoding the sequence of mRNA codons. So we will decode this mRNA, and that is the process of translation, and we'll ultimately be making proteins out of it. All right, so you need the other 2 types of RNA now. You have the mRNA that you made in the nucleus, and now we have transfer RNA. So transfer RNA, let's move my face. Transfer RNA is this guy here. Okay, so we have a start codon right here, so that's where transfer RNA will first come in. There's methionine, all right, AUG is methionine there, and transfer RNA will bring in this amino acid, and this blue part here is our ribosomal RNA. See the ribosome? All right, and that ribosome is free floating in the cytoplasm, and mRNA is sort of like the, driving to the ribosome like a bus stop, and then the transfer RNA is like dropping something off at the bus stop and then leaving. Dropping a kid off at the bus stop and leaving, and you can pretend like these little amino acids are your kids. I don't know. Come up with something to help you remember this process. So translation starts when a ribosome attaches to the mRNA molecule. All right, so here's the ribosome attaching to the mRNA molecule. Then the tRNA molecules carry the amino acids with them and bind to the mRNA codon. All right, there's the tRNA. And we call this an anticodon. This is the codon. This is an anticodon that the tRNA carries in. and it just finds those amino acids in the cytoplasm and brings them in. All right, so translation is the polypeptide assembly. So we talked about what peptides were. They're one of the 4 macromolecules. They're proteins, right? So the ribosome helps form a peptide bond, and it breaks the bond holding the first tRNA molecule to its amino acid. So then it breaks it and it goes away, and what you're left with are these chains of amino acids. Okay, the tRNA will drop it off and leave. The ribosome reaches a stop codon eventually, and that releases the new synthesized polypeptide. So this polypeptide molecule is completed, and that is the end of translation. There's your stop. And there's your polypeptide up top, right there. The roles of RNA in translation. So all 3 major forms of RNA, mRNA, tRNA, and rRNA, are involved in the process of translation. Once again, messenger carries instructions for the polypeptide synthesis from the nucleus to the ribosomes in the cytoplasm. tRNA carries amino acids to the ribosome and matches them to the coded mRNA message. And then the ribosomal RNA forms an important part of both subunits of the ribosome. All right, so here is the whole process once again. We've got transcription inside of that nucleus making the mRNA strand. That mRNA strand is carrying the codons that it made from DNA. Okay, and then it goes and finds a ribosome. And then those tRNAs come in with its anticodon, match up the proper amino acids that are needed for the polypeptide to make whatever protein your cells and body needs. The central dogma of molecular biology is that information is transferred from DNA to RNA to protein. So when I say central dogma, you think of DNA to RNA to proteins. Okay, gene expression. When a gene segment of DNA code is used to build a protein, scientists say that that gene has been expressed. So these different proteins express your genes for you. All right. And lesson 3 is about gene regulation and expression. A lot of this is extra information. I'm going to just go ahead and go through the PowerPoint here, but I would not expect that you need to know much of this, just a few words, and we'll talk about that. Okay, so first it talks about prokaryotic gene regulation. So there are things called DNA binding proteins in prokaryotes that help regulate genes by controlling transcription. One of the keys to gene transcription in bacteria is the organization of genes into operons. All right, so you don't really need to know this, unfortunately, but I'm just going to, again, briefly go through it. So when lactose is not present, they have something called lac genes that are turned off by regulatory proteins that bind to the DNA and block transcription. So bacteria don't always need to be evolving, so if that's the case, they will block transcription from happening. All right, so promoters and operators are still words that you should know, just in general. We're still talking about prokaryotes here, but located in the front of the operons, 3 genes are 2 regulatory regions. So you've got a promoter here and an operator here. All right, so this is where the operator repressor will bind. And then you've got this thing called the lac repressor gene. And then you've got the lac genes here. Okay, so the lac repressor is important because it blocks transcription from happening. When the lac repressor binds to this area here, okay, the RNA polymerase cannot reach the lac genes to begin transcription. So that is a repressor protein. All right, so there is a lactose right here. When lactose is added to the medium, it diffuses into the cell and attaches to the lac repressor. So lactose is needed in order for proteins to stop and to repress the proteins that are being made. All right, and it still has RNA polymerase being used. It still uses mRNA for transcription. and then translation has to happen. And it's just a very simple process because they're just bacteria. So eukaryotic gene regulation is important. The only thing that we really need to know from this lesson is Tata box. A typical eukaryotic gene has a Tata box. What is that? All right, so this is the Tata box right here, and it allows for our prokaryotes transcription to actually happen. All right, so you have RNA polymerase here, and it helps to direct the Tata box and actually proceed with transcription. All right, so transcription factors. By binding DNA sequences in the regulatory regions of the eukaryotic genes, transcription factors control the expression of those genes. So what do transcription factors do? They control the expression of the genes in a prokaryotic cell. Okay, that is another word that you should just know the definition of, transcription factor. Cell specialization. So complex gene regulation in eukaryotes is what makes differentiation and specialization possible. All right, again, these are not heavily assessed. I don't even think it's a standard, but because it's in the book and already in this PowerPoint, I'm just going to keep going through it. Okay, so regulating gene expression, this is important in shaping how a multicellular organism will actually develop. Each of the specialized cell types found in the adult originates from the same fertilized egg cell. So if you get anything out of that, just understand that genes are turned on and off, that expression happens, and in prokaryotes, it's a very simple process, but whereas eukaryotes, you saw it was pretty complicated with lac operons needed, and it's all about just gene expression, how those traits are being expressed. So the only reason I showed you this lesson is because of these words here. These are called homeotic, homeobox, and Hox genes. Okay, so these are vocab words that you should know. Homeotic genes are the ones that regulate organ development. Homeobox genes code for transcription factors, so they help determine which genes are going to be expressed. And Hox genes determine the identities of each body segment. So Hox genes are pretty cool. They help to determine, like on a fruit fly embryo, what develops into the wings, where the wings attach, what develops into the cephalothorax or the thorax. And then same thing with an adult mouse, like where is the tail going to go? Without Hox genes, there could be a wing coming out of the forehead of an animal or whatever. So Hox genes are what determine the body segments. And this slide here is really the only reason I went through this lesson. Okay, so epigenetic mechanisms control which genes are on and which are off during each stage of an insect's life cycle. So another vocab word for you, just to understand the stages of a life cycle. For example, a pupa to larva to adult, butterflies, insects. Okay. There are environmental influences, and we keep talking about this, like how traits are expressed, and the environment does have a big role in traits that are expressed. So things that can affect gene regulation. For example, this rabbit was raised at temperatures below 25 degrees Celsius, and because of those lower temperatures in the environment, this bunny comes out with extremities that are darker. And raised at temperatures above 30 degrees Celsius, this rabbit is all white. So that is a common thing we'll see in the environment with not just mammals, insects. It's a very common thing. All right, lesson 4, mutations, is a pretty intense lesson, and we will be doing a couple worksheets to go along with this. So there are types of mutations that we need to talk about. So mutations are heritable changes in genetic information. Mutations fall into 2 basic categories. We've got gene mutations and chromosomal mutations. So gene mutations and are a type of point mutation. So a point mutation is a change in a single nucleotide. There are three main types of point mutations that we're going to talk about. So before mutations, this would be your normal segment here. This one is showing you a substitution. So the C gets removed and a T gets put in, and then that amino acid becomes histidine instead. And over here, we've got an insertion. All right, where A gets put in there, and serine is now replaced, and also tyrosine and leucine end up getting messed up in the future. So these were all different amino acids compared to the original. And if deletion happens, codons can't be completed, so the amino acid at the end completely doesn't form. So the polypeptide will completely be broken. And obviously that can cause serious issues. Okay, so point mutation, again, type is substitution. So in a substitution, one base is changed to a different base, like we said. All right, so this one was changed from arginine to histidine. There's arginine, there's histidine. All right, a point mutation is insertions and deletion. So point mutation is what we're going to talk about here. So insertion is when a single extra base is added into the code, like we talked about earlier. Okay, and we have these three amino acids that are changed now. And then deletion is when a single base is removed from the code. Okay, so here's the other type of mutations, chromosomal. So chromosomal mutations typically will happen during mitosis and meiosis, and you'll see deletion, duplication, inversion, and translocation in this chapter. So the original chromosome looks like that. Sorry guys, it's been a long day, I'm super tired. Deletion would cause, you know, a segment to be deleted. It's pretty obvious. Duplication, all these words pretty much mean and do what you understand them to do. Duplicate, okay. Inversion, something switches, like so a segment on the chromosome switches, and then translocation would be an extra piece of chromosome being put in. Okay, so this G and H gets translocated to here. Okay. The effects of mutations. So mutations can be helpful, actually, and they're not always harmful, or they can sometimes have no effect on an organism at all. Some mutations arise from mutagens, which are chemical or physical agents in the environment, like UV, for example, UVA, UVB. Some of the most harmful mutations are those that dramatically change protein structure and the gene's activities. This here is showing you sickle cell. So an example is sickle cell disease. It affects the shape of the red blood cells. A normal red blood cell has a circular-ish oval shape, where sickle cells are kind of pointy and look like a crescent moon. And if that's the case, then they clump together, they cause clotting issues, and they can't carry hemoglobin effectively, meaning there's not enough oxygen getting transferred throughout your body and to your brain, so you have extreme fatigue, etc. All right, so the effects of mutations that are beneficial mutations often will produce proteins with new or altered functioning that can be useful in an organism in a changing environment. So the real big benefit to a lot of organisms would be an evolutionary standpoint. And that concludes our lecture for today. Please let me know if you have any questions, and I will see you tomorrow. Bye, guys.